Yeah, it works.
Oh.
All works. All works.
Okay. Thank you, Rodrigo, and thank you, Alexander. Thank you very much. I'm a computer scientist. 30 years ago, I was a computer scientist, and I will try to connect to computer science folks, but I have not done computer science in a long time, as you can see by my ability to get my keynote working here. I've always been a rebel, and so I got a Macintosh many years ago, and I've never looked back. I never owned a PC except when I had to do some stimulus presentation. So Unix and Mac, that's it. So anyway, thank you so much. Thanks, Marcella. Thank you, Rodrigo. Thanks to the organizers of the conference. It's a real honor to be here. I have never been in Chile before. I've been in Buenos Aires once. So this is my second visit to South America. Despite the fact that my wife is Mexicana, my Espanol is really horrible, and I apologize for that. If I speak too quickly, stop me. If I say something that you can't understand, tell me, and I'll slow down, I'll stop. I could try French. Could try a little Espanol, but not much. So anyway, I'm going to talk about my work and what I believe is the future of the neurobiology of language, which is a field that I've been embracing for about 10 years. Started a society for neurobiology of language and started a journal on neurobiology of language, and really embracing a new approach. I think it's a new approach to the study of the language and the brain, and that's what I will talk about. Thank you all for participating. The outline, I have the talk will be in two parts. One, I'll talk a little about principles that are guiding my work, and next I'll show you some examples. I chose principles and examples that I thought would be relevant to computation. So although I do not do computer science now, I do understand some computer science principles. I'll try to relate some computer science along the way as we go. And in the last half, I'll actually talk about data that is not yet published. I'll talk about where we're going in my laboratory with this kind of work. So first, the brain is a computer, and this is fun. We'll see what you think. If you'd like to interrupt me with questions, that's fine. If you'd like to wait till the end, that's fine too, whatever you would like. I'm happy to take questions. I'm also happy to take criticisms and arguments from the floor. I love Latin America, and I love Southern Europe, and I know we can have some good debate. So the digital computer is not so old. It's not even 100 years old yet, as you know. This at the bottom is a little schematic of what a serial digital computer looks like. Whether you have a Mac like I do or a PC, you have an input device, a CPU, an output device, and some memory. And that's about what a digital computer is. And that's the 218 computer. This is the computer I used in high school, was this thing. And you could do programming by toggling the switches on the front. But you had to get the octal right, or it would line by line. One set of switches, enter. One set of switches, enter, and that's how you wrote a program on the, that's a Digital Equipment Corporation PDP-8. We've now come a long way, and now we talk about distributed computers. And I call this type of computer the all-action, no-talk distributed computer. This distributed computer was the early invention of this was on naval ships, because they wanted naval ships to communicate with each other and to work together. And so this is the kind of, the origin was in multiple computers in multiple places working together. This is sort of like how a lot of internet kinds of computing works now. That you have a big computer that can do a lot of chunking, and another big computer that can do a lot of chunking, but it's very expensive to communicate. And that's why I call it the all action, no talk. Communication is expensive, processing is cheap. And of course, in cognitive science, which is our field, that kind of computer has become a metaphor for a lot of processing. And this is one from a friend of mine, Tim Challis, who's a neuropsychologist, old neuropsychologist now. But you see models like this in psychology all the time. And I think it comes from the idea of the all-action, no-talk computer. And the computer metaphor has really influenced cognitive science in a big way. Here's an example of from Neisser, Ulrich Neisser on cognition. Here's an example from Fodor and Chomsky on language. You have computer A, computer B does its processing, computer B does its processing, computer C does its processing, and so forth. And it's come to neuroscience. So now we have the idea that we have a talking node, a talking process, a reading process, understanding process, social communication process, and they're big computing elements that all communicate with each other. So the question is this the kind of computer that the brain is? And I don't think so. I think the brain is a massively distributed computer. I call this the all talk, no action computer, because every node in this kind of computer can do a very, very minimal function. But the power comes from the massive communication you get between the nodes. And so if you can do, if you have an adder or a multiplier, some very simple kind of process, if you have a large interconnected system like that, you can do very, very advanced computing. And that's what I view the brain as like. And the human cerebral cortex, which has about 30 billion neurons, 100,000 kilometer of axons, computes in this way, where each node doesn't do very much. It does a little summation, not too much. So how does the brain work? Well, is it a plumbing system? That's about 122 years old now. Is it a telephone switchboard? That was really common for a long time. Logic machine, you've seen that. Digital computer, of course. Hologram, constraint satisfaction device, connection network, set of large brain regions, or a massively distributed system. And I say now we're taking the massively distributed system and we're going one step further and we're going into a complex substructure within the complex structure. So we have an organization within the complex structure that makes up this model of the brain. So we have nodes that are very, very influential, hub nodes. We have core nodes, and we have periphery nodes, and we have nodes in between. And that's and using that kind of structure, you can explain to some degree both lesion data from patients who have damage to their brain, as well as normal function. So let's get into what's language and what's neurobiology. So this is traditionally, any of you in the room study language in the brain besides Marcella? Nobody? Oh. Giovanna does? You don't? Okay. Okay. So the language is a social product of the faculty of speech and a collection of necessary conventions adopted by a social body to enable individuals to exercise that faculty. That's from de Saussure, and I'm very proud de Saussure is in my neurolinguist tree some years ago. What is language use? Communication between people using language aimed at understanding each other with a purpose in time and space. So language is not something that exists without having people use it. So the dichotomy that was presented in the 20th century about language competence and language performance, I don't really buy it. I think that we need to study language in the context of its use. And ecologic language is language used by people at a time and place, aiming to understand and to be understood, fulfilling a purpose. And that's the language I want to be studying. That's what I view as the object of inquiry. So neurobiology, so we have a sort of a continuum from cognitive science, symbolic processing. The brain does process symbols, but they're not implemented. You folks are, many of you in computer science. So the difference between the output and the implementation is clear. The implementation is not symbolic, but we need to do symbol processing, and we can study symbol processing. Cognitive neuroscience is a branch of experimental psychology that uses brain imaging to understand psychological phenomena. You want to understand memory better? You can do an imaging experiment, will help you understand memory better. You want to understand language, you want to understand attention, you can do an imaging study, you can do an EEG study. So to me, cognitive neuroscience is a branch of psychology. And neurobiology of cognition is different, and that's what I'm trying to move our field, neurobiology of language, toward. That's what my advocacy is about, to get us trying to think about what for me, as a computer science trainee, it's the implementation details. I care about the implementation details. And I think to study language and the brain properly, we need to care about the implementation details. And I wrote this little short one-page, two-page paper a long time ago when I was at the University of Pittsburgh. It was a commentary in behavioral brain sciences where I said the real functional architecture is gray, wet, and slippery. So I've been doing that for since the time that my kids were born, and probably some of you. So neurobiology, though, depends on animal models. So how do you have what's an animal model for language? What do we do? Is it a bird? No. The bird talks, but the bird doesn't have language. So what is an animal model for language? And that is a little too loud. Let me try it again.
Yo no hablo español.
I think I need Alex again. Alex, can you turn it down? Oh, you're here.
Yeah, I'm here.
I tried to turn it down here, but it's not turning down. Let's see. You think that'll do it?
Yeah, it works. You need to return to the past audio, or just load
I don't speak English either. We got it.
Yeah, we got it.
Gracias. So this is not a great animal model, right? And back when I first started in this field, I used to be invited to give lectures in psychology departments and neuroscience departments. And the neuroscientists, the first thing they asked me is, what's my animal model? They wanted me to discuss cellular molecular biology. And for, 5 or 10 years, I did not give lectures in neuroscience departments on language. I talked about motor system because they're very, highly coupled, and I was doing research in both. So we need animal models for all levels of biological research. But nowadays, but the question now is with MRI, we can actually do physiology. We can understand true physiology because of the advent of high-resolution brain imaging. So when I went to those neuroscience departments, they were upset because I couldn't do real physiology. Describe a patient who has a language disorder because of a stroke, have language problems, and I can explain the language, describe the language problems, but I couldn't explain the physiology. And I was asked on my first faculty job, could you give a lecture on physiology of language? I said, I'll give a lecture on language, but I can't talk about physiology. Now we can do physiology in human beings, and that's new. That's only for the last 20, 30 years that we can do physiology in human beings, and it's a very exciting era. And so now we have animal model of human neurobiology. And the animal model we use most frequently, of course, is the undergraduate student. And that's our animal model that we use because we have these imaging devices, and so we can test this. Of course, the original animal model has now been extended, and we have more recent models, typically aging adults, adolescents, children, and we can do a lot of real physiology in human beings. So we now, human subjects research can now be truly neurobiological in nature, and that's new. That's not been true for a long time. That is quite a new phenomenon. And when people ask me, are you doing clinical research or basic science research? I say, I'm doing basic science research. 25 years ago, if you were using humans, you were not doing basic science. They would not believe you were doing basic science. Now we can do basic science. So let me challenge the room with some assumptions that guide my work, which is assumptions that I think we need to revisit. So localization is one. How many of you have studied brain function through looking at what regions do what? That's probably part of every single course you take, is this region does this region does this region does this. And many of you, if you're in computer science, that's probably what you've been taught to believe, is that different regions of the brain do different things. The mapping from brain structure to function is not one-to-one, however. It's many to many, and it's very complicated. And we need computer science to help with this. Ecology, the study of language processing in the world, is required to understand brain organization. That is to say, studying language as it's actually used is the way we need to go about studying it, because if you do artificial experiments in language, for example, push a button if this sentence makes sense to you or not, okay? That's a great experiment to do in a psychology laboratory, because the dependent measure in a psychology laboratory is an error rate or a time. So you can say, oh, this one is hard to understand because it took longer. Or this one is easy to understand because it was shorter. This one's not grammatical, so it was harder to understand. The problem is that when the dependent measure is what the whole brain is doing, then that's not a good study, because when you do that, you're going to get the whole brain activity for making a decision, deciding whether something is grammatical, making all sorts of decisions. And that's what you'll see in the brain image. I'll see brain imaging of decision making. And in our field, we call that metalinguistics, meta-language. So we're studying the processes to decide about language. We're not studying language. So I'm advocating that we study language. Furthermore, I went through my argument for the brain as a massively distributed computer. We need to understand the brain. We need to have explanations of the brain in terms of distributed computing and massively distributed computing with organization. And then time and space. I'm guilty most of my career for doing fMRI, functional magnetic resonance imaging. Does everyone know what that is? Anyone not know what that is? Okay. Functional magnetic resonance, good. Functional magnetic resonance imaging is a relatively recent technique. It's only about 20 years old now, 25 years old maybe. And you can use an MRI scanner, which is a scanner that's used in the hospital to diagnose brain disease. And you can use that scanner, change the algorithms in the scanner to be sensitive to blood flow. So you change the algorithms, instead of being sensitive to abnormal tissue in the brain, which is what the neurologists, neurosurgeons want to do, you change the algorithms in the computer, in the MRI scanner, which has a large computer as part of it. You change the algorithms to be sensitive to blood flow. And it's known that when you do a task, when you do some sort of a task, the blood flow will increase to the part of the brain that's involved in doing that task. So when you see an fMRI image, and I'll show you some, what you're seeing is you're seeing a color map that shows very bright colors, meaning the brain is very active during a task performance, or dark colors, meaning the brain is either inactive or not active during a task performance. Originally, I did it when I first was testing out my algorithms. I did this. And those of you who have studied any neuroscience or even read the newspaper know that when I move my left hand, the right part of my brain ought to get active. And in fact, the part of my right brain that's active, there's a motor region of the brain that gets active. I said I don't believe in one-to-one mapping, so in fact, there's a lot of regions that get active, but the motor region of the brain gets more active, okay? And that's the substructure on the system. So that's fMRI. And I've done a lot of fMRI, and I've shown in my fMRI studies where certain things are happening. That's the space description of the brain. So I'm explaining the brain function in terms of space, where things happen. But I'm not explaining when they happen or how they happen. And so the argument is we need space and time to do that. And that takes complex mathematics. So the reason we think that regions do specific things is because for 100 years, the only way to study the brain was through strokes and other disease. So if you have a stroke, part of your brain is dead. And then if I look at you and say, oh, I have a part of my brain that's dead, what can't I do? What function am I missing? Then I can say that part of the brain did that function. And that's how we studied the brain for 100 years. And so we have the legacy of holes in the brain, and it's led to these are functional MRI images. And these are MRI images for different kinds of very complicated tasks. But what the designers of these studies have done is to say, they say, I want a location. So by using thresholding methods, they say, I'm only going to look at brain activity at a certain level. That's like saying, okay, I'm only going to look at South America through the tips of the Andes Mountains, okay? Because I'm only going to look at the highest points in the terrain. But the brain activity is a terrain. The brain is always active. There's a lot of activity. Here I'm just looking at the mountaintops. And the mountaintops are not necessarily reflective of what's going on. Particularly when I say this is an average of 20 people. Because this paper there on the bottom, we showed that when you do an average of 20 people and you get a spot like that, it's very, very much the case that no individual has activity in that spot. Because they have a lower, they have a small hill, not a mountain, and they average together small hills from everybody, and you get activity, whereas the big hills are elsewhere in each individual. We can go over that later if someone is interested. So here is a reading network. The purple nodes and the blue nodes constitute, for example, it's a Gedanken experiment. The blue nodes and the purple nodes represent reading. Oh, and now the blue nodes and the purple nodes represent talking. So what's the function of the purple nodes? Talking, reading, or something else? And this is how the brain, this is from Randy McIntosh. This is how the brain works. Randy McIntosh is a professor. He was at Toronto. He's now in Vancouver, British Columbia, and a very, very smart person. So when I put my first people, I put them in the MRI scanner to listen to real stories and real movies. This is what I got. So remember, bright colors are activity, and dark colors are decreased activity or lack of activity. So what do you see? Do you see activity in a single spot? No, you see activity throughout the brain. And that's natural comprehension. That's what you're doing when you're actually in the world. I can't be involved without really trying hard to speak with people without seeing them, without being in an environment, without being in a place. Language is processed in the world ecologically. And that's what you get. And understanding that is difficult. And this just reinforces that point. People understand better in context. They need expectations, previous knowledge, experience, and so forth and so on. So the approach that I'm recommending, as you know, is brain connectivity. This is brain connectivity is not new. But it's been refined and becoming better and better. And the picture on the bottom is an example from one of the best imaging groups. And you can see lots and lots of different nodes and connections. The top is an example of the connections that exist in the human brain. Those are the hardwired connections. Those are hardwired connections. That's the axons, the myelin that connects different regions in structural terms. So here's just another example of the connectivity of the brain. That's from Desjardins, who wrote about this in about 100 years ago, maybe 1919, something like that. Oh no, 1895, even more than 100 years ago. And then this is recent imaging, structural imaging of connections. And that's called connections occur in the white matter of the brain. The brain has gray matter around the cortex. And then in between is a whole bunch of connections. And it looks white pathologically. So there's various ways to do connectivity. You can look at networks in a static form. And some of you in computer science probably study graph theory. We use graph theory. And we use, we look at modularity. We look at integration, segregation, and various forms of graphs. Interestingly, if you take an image of the brain, and an image of the brain with fMRI, I didn't tell you this, but the image of the brain with fMRI, you have to look at the brain over a long period of time. So you have an image about every second for an hour. And so you have a different image all the time, and you can see changes in what happens in those images over time. So if I'm moving my hand, and then I stop, and then I'm moving my hand, and then I stop, I'll have activity for a while, and then I'll have no activity. I'm talking about in the motor system. Then I'll have activity, and then I'll have no activity. So this changes over time. This is an example. If you look at everything on the y-axis is a region of the brain. Everything on the x-axis is the same set of regions in the brain. These are the connections between them at one time point. And but if I look over time, this is real MRI, fMRI taken over time. And this is actually someone who's not doing a task, just at rest. And you can see that the connection pattern changes over the course of this. I think it's about 2 minutes, 3 minutes. So over the course of 3 minutes, the resting state of the brain, the connection pattern is changing all the time. And that we use that to try to understand what's going on. And there's many spatiotemporal scales for investigation in the neurobiology of language, from very, very, some cellular function. And we can do some cellular neuroscience of language now because of diseases like Alzheimer's disease, which is related to a certain protein. Frontotemporal dementia is related to another protein. So we can do some cellular biology of language all the way up to behavior. So finally, for those of you interested in combinatorics, I mentioned the cerebral cortex has 30 billion neurons. And suppose you have 5 brain regions. What's the connectivity? What's the complexity? What's the computational complexity of this, of building networks with only 5 nodes? And this also applies to genes. If you're interested in genetics, it also applies to 5 genes. So the combinatorics here are, and this is Duke is the one who pointed this out. He's a physicist by training, that as you get more nodes, look at the number of possible networks you can have. And it just grows exponentially. This is a million different networks with 5 nodes. We said there's 30 billion neurons in the brain. And with our models, when we make models, we sometimes look at different regions. We say, how are the regions connected to each other? And so the computational explosion is enormous here. So just with 16 brain regions, it becomes ridiculous. And here's an example of what we actually use today. We use 80 regions frequently. My work for 20 years used these 80 regions. Some people now have divided all this into subregions, and they do 1,000 regions per hemisphere. So now we're trying to understand connectivity patterns during processing of language with 2,000 different regions all connected in certain ways. Now we can constrain the connections based on anatomy a little bit. Some things just are not interacting with each other. But in general, this is an enormous computational problem. If any of you guys are interested in large data sets and major league complexity problems, this is it. I'll put that in for y'all. Y'all is a word that we invented in Texas. It's the second person plural of you in Texas. Okay. To do this stuff, we have to use high-performance computers because it's impossible. And even with high-performance computers, we're running into enormous amounts of difficulty. And thankfully, the National Science Foundation in America has contributed to this. The Texas has a really wonderful advanced computing center. And we've done some work in parallel workflows over many years now. That one's published more than 10 years ago. Okay. So what do I do with all this stuff here? What do I do with all this information? Well, I have tried to do research in this vein, and some of it succeeds at meeting my assumptions, and some of it doesn't. But I'll show you what we've been trying to do. So we first started with Aesop's fables. These are famous stories for children, although nowadays people don't like giving them to children. They're sometimes a little violent and stuff. But
an ant was running around in the sunshine looking for food when he saw a caterpillar's cocoon hanging from a branch with something inside of it. The cocoon wiggled a little, and the ant realized that something alive was in there. Poor animal, cried the ant. An ant was running around in the sunshine looking for food when he saw a caterpillar's cocoon hanging from a branch with something inside of it. The cocoon wiggled a little, and the ant realized that something alive was in there. Poor animal, cried the ant. What a horrible...
So you get the idea. In one case, we have no... there should have been a static picture of the face, but in one case, there's no movement of the lips and mouth. The other case, there is movement of the lips and mouth. And when you speak and when you understand speech, you're looking at the lips and mouth, even with normal hearing. With abnormal hearing, it's crucial. But even with normal hearing, you frequently look at the lips and mouth. And here we did a study. There were two other conditions, which I won't show you today. There was another condition where she was allowed to gesture. And there was a fourth condition where she made stupid gestures, gestures that were not related to the speech. So you see me talking and I'm moving my hands like this. Suppose I was talking and I was doing this kind of stuff. Those are the kind of gestures that are in the control condition. So we had just the static, we had lips and mouth, we had normal gestures, and then we had stupid gestures that don't relate to the speech. What's that?
No, our former president made this.
He does those? Oh, okay. It's hard to understand that stuff. I mean, we did some studies looking at how that would inhibit, actually. It inhibits comprehension. Let me see if I can go forward. Yeah, I can. So when you compare audio visual to audio only, you have, there are differences in brain activity. So audio visual, even in areas that would thought to be more language-oriented areas, auditory areas. So in particular, the frontal lobe, you see in audio, I know well enough not to talk when I'm not at the microphone, but you can see there's differences there in audio versus audiovisual. And what we did in order to try to follow the outline I mentioned to you, we tried to build a model of the connectivity of the different regions with each other. These are called effective connectivity models. One example of an effective connectivity approach, for those of you who know statistics at all, is structural equation modeling, is one kind of approach that's used in effective connectivity. And these graphs are quite complicated to interpret. It's a lot easier to say there's activity in this front or there's activity in the back. Here was how do we describe what did we find? And for years we couldn't publish stuff, and now we can, but it took a while. So here's one way to summarize the findings there. These are brain regions, premotor cortex ventral, premotor cortex dorsal. That's there's the motor cortex executes the function, is the final common pathway in the cortex, and then right in front of the final pathway are areas that do motor planning and some aspects of execution. And then superior temporal lobe is part of the brain on the side that is typically involved in things like comprehension. And so if you look at the effective connectivity, it looks like the ventral part of premotor and the posterior supertemporal are more sensitive to the mouth. And when we did it with the manual gesture, we found different regions were sensitive to manual gesture. So perceiving the hands and perceiving the mouth led to different activation patterns. We did the same in development. The brain changes a lot over the course of development, as you know, and with input. And we thought we used Aesop's fables because they could be used in children and adults. So comparing children and adults on the same task, the same fables, we have differences. There's more frontal activity in adults. The adult brain, the child's brain has a gradient of development where the highest order structures, the parts of the brain that are involved in the most advanced kind of reasoning, develop later than other regions. And here we have more activity in the front and more involvement there. But again, how do we explain what's going on? We don't just want to show a pretty picture. We want to try to model this. Again, this is effective connectivity. And to try to understand that better, we looked at which nodes were the most important in explaining the differences. And if you compare adults versus children, there's two that really strike out, and one of them really highlights the role of the frontal lobe, which develops late, the role of the frontal lobe in these tasks in adults compared to children. We've done the same in aphasia. Well, Susan Duncan did. She was my PhD student. She's now at Louisiana State University in the United States. And we did a study, an aphasia study, language disorder. So aphasia is a word for a language disorder after a stroke. So if you have a stroke and you can't speak or understand, we call that aphasia. We estimated this morning, we had a couple of speech therapists this morning at my talk at the medical school, and there are about 1 million people with aphasia in the United States. We estimated there are probably 80 or 90,000 in Chile with aphasia, chronic aphasia. And so we tried to, this is a study to do therapy, to try to help those people regain language skill. And we did an imitation-based therapy. It's called action observation therapy, if you want to look it up. It's been used in motor system recovery, Parkinson's disease, other things. But you observe actions and try to take in what you've learned from observing those actions into your own action profile. So trying to learn from that. You can stand up, it's all right. So Susan did this. We had some beneficial effects. Not overwhelming. I mean, the field of aphasia therapy is very difficult. We have not made tremendous inroads. But we did a lot of imaging. And one of the things we did in imaging is we expected, we built these models, these structural equation models, and we expected people who responded to the therapy would have networks that resembled more normal people's networks. So we had a bunch of normal people's networks observing and imitating speech. We had a bunch of people with language disorders observing and imitating speech. And we thought that the people who succeeded at therapy would be more like the normals. That's not true. And when I first presented that at a rehabilitation hospital, they told me I was wrong. And the reason I was wrong is because the injured brain compensates for damage. So it compensates in different ways, and everybody would compensate potentially in a different way. And here, it turned out that what the story was that Priya Santhanam came up with is that if you have a longer-term injury, then you're more likely to differ from the normative model in your recovery. So if you had injury 5, 10 years ago, then you may have organization on a part of the brain that doesn't normally do language. Whereas if you have injury in the medium term, you're more likely to have reorganization near where the stroke was, on the area around the stroke, the parts of the brain that are involved there. And that led to, I guess there's 2 articles on that. All of these are published, so I can send them to anybody who cares, would like these. In the brain, as I mentioned earlier, there is the brain at rest goes through a lot of different states. And when I showed you the different states of that one subject, that was about 3 minutes worth of resting state. And you saw the connectivity change all the time. And that, the folks at Washington University in St. Louis have demonstrated that there are some subnetworks that occur in this resting state. So in the resting state, there are certain regions that hang together during the rest. And we found the same is true in people with aphasia. And using dynamic functional connectivity, which is what I showed you, that's how the connectivity changes over time, we actually found that patients who recovered from aphasia, they actually stayed in one of these states longer than other people. So there's functional connections, they change over time, but there's regularities. So suppose I have an hour's worth of resting state, for example. During that hour, people may tend to have patterns of connectivity that fall into different groups. And you can, by using k-means clustering, other clustering algorithms, you can come up with regularities in these states. So the patients are in maybe, I think she found 10 different states of connectivity the people were in. It turns out if they were in one of these, they were more likely to benefit from therapy. So this is an initiation into something called precision medicine. Have you ever, people heard of precision medicine? That's when this is a new way of doing medical care where I take an individual's characteristics, genetics, physiology, and I make a therapy specific to that individual. It's very important in cancer now because there are certain cancer drugs that only work on certain kinds of genetic makeups. So this is called precision medicine. That's what we're trying to do in language in the brain. So finally, what we're doing now, and this is kind of, this is the latest work that we've done. This work is done by Chi-Ming Ting, who's a professor at Monash University in Malaysia. This is work, this is advanced statistics. And if any of you are interested in matrix decomposition, advanced linear algebra, this is the guy you should talk to because he's the one who's applying this to imaging data very well. And what we did is we tried, I told you I want to understand how the brain works in both space and time. And he developed an algorithm, it's called low sample sparse decomposition, matrix decomposition. And he used that to analyze a movie. I'll show you a little bit of the movie, just because it's fun. 13 subjects watched this movie. The movie's called Are You Smarter Than a Fifth Grader? And the premise is adults come in and they're contestants in a TV show to win a prize, and they have to compete against 10-year-old children. And if you know 10-year-old children, they're a lot smarter than some adults in some topics. And it's a very funny show.
And we're giving grown-ups the chance to win $1 million.
So we image people watching this TV show. Okay? We had them in the scanner watching this TV show in the MRI scanner. And so you can see there's a lot of activity going on. There's a lot of noise. There's a lot of action. And this is the contestant. And I'll just get through her introduction because it's very funny.
There's a picture of Megan in the 5th grade. What kind of student were you?
I was a teacher's pet. I was a big nerd.
What are you majoring in grad school?
I am studying neuroscience.
Like brain.
Yes.
Oh, wow.
Yeah.
Going for your PhD, I understand.
Yes, I am.
So my collaborator, Jeremy Skipper, who's at University College London, he's the one who did the imaging. And he's my former PhD student at University of Chicago. And so he put these undergrads, University College London undergrads in the scanner, played them this TV show, and then what we tried to do is try to figure out in space and time what's going on in the people's brains who watch the show. To do that, we had to take the MRI. Has anyone seen MRI data? A couple of you. MRI data from has anyone seen EEG data? Anyone seen time series data of any sort? Physics time series? Yeah. So these are time series. The sampling rate is slow. The sampling rate on fMRI is a second or two. So we have time series. If we have the brain, and we divide the brain into voxels, like you all know from computer vision school, we divide the brain into voxels, there may be 256 by 256 by 50 voxels in an average brain, the way we slice it. So you look at these 100,000 or so voxels, and each one changes over time. The activity in that changes over the course of the movie. And what we need to do this kind of imaging is need to say at every point in the show what's going on. And in this particular case, I'll show you what we coded. If I can find my pointer. Oh, there it is. Oh no, this way. So we coded here. So we coded in the show, when did noise occur without speech? When did music occur? When was there speech and silence? When was there speech in noise and silence? And you can see this is a rich stimulus. It's loud, boisterous. It's a very rich stimulus. So we divided into those 5 categories, and we said, Chi-Ming, can you distinguish the states that are relevant for that? And so he did. He used this fused LS approach, and this is I think one time point, and here is the connectivity in 13 subjects at that one time point. Okay, so one time point, that's the connectivity matrix for each of those 13 subjects. And by using his low-rank, sparse decomposition, he found that there's commonalities in all the subjects in that. And these that are the residual, noise matrices. And if you look here, you see that the colors are when he decided that the algorithm found noise. And then the gray, which you can see around it, is when noise actually occurred. So you can see this is a pretty accurate model of when these different things occurred in the movie. And this is silence, and you can see that the green is silence. And you can see the gray, it's a pretty good predictor. The green being the model, and the gray being the actual result. And so we found that through this method that there's higher functional connectivity in regions involved in speech perception and language comprehension for speech and noise compared to speech, and we have a couple of pathways, and increase in connectivity between auditory and premotor, motor, and somatosensory regions in response to speech mixed with noise. So we did a spatio-temporal analysis of 13 undergraduate students watching this TV show. So just for fun, this is my, these are social emotional networks in the brain, in the brain of the macaque. I didn't realize I'd be studying social and emotional networks, but the brain processes social and emotional information, and there are networks that are involved in doing that. I thought I was going to study language. However, let me show you. Here's some of the biology of this. Emotional valence and emotional processing. And I was going to get to the punchline. I'm going to get to the punchline. This lady, I'm going to get the punchline, go back. Annette Glotfelty came to my lab. She said, I want to work with you. I said, okay, I want you to work on noun phrases, right? Very interesting linguistic topic. You know what a noun phrase is? The big man, the happy boy, el professore di neurologia. Sorry, these are noun phrases. I said, you want to study noun phrases? And she came back a month later and she said, no, I want to study curse words, okay, taboo words, right? And, I said, sure. And so I realized when she started saying that we are talking about now this social and emotional processing networks. What makes a curse word a curse word? Why do you perceive it the way you perceive it? What are the brain networks that are involved in perceiving a curse word? And so, we get into all of these, the animal literature, and then human literature and emotional valence and arousal. Are curse words, are they curse words because they have arousal? Do they have emotional valence? What is it that makes a curse word a curse word? And we went to Jeremy Skipper, my former student. If any of you are interested in working on data like movies, Jeremy published the entire data set in an online repository. And there's a paper that describes how to access it and where it is. It's in Nature Scientific Data, and you can use these data. So we went to this. Jeremy had 86 participants watching full movies. So he graduated from TV shows to full movies. And one of them is this Pulp Fiction, which have you ever seen Pulp Fiction? Yeah, I mean, it's violent, and there's a curse word every second in this movie. So I was worried, actually, about attenuation of effect. When the brain hears the same thing over and over, there's attenuation, a considerable attenuation. That's why when your grandmother uses a curse word, it means a lot more than when your buddy uses a curse word, because it's all contextually determined. So we did that, and Annette found that 7 of the movies had enough curse words to be able to be studied, and there were 56 participants left. So she's trying to come up with still what makes a curse word a curse word. And what we've come up so far is arousal and valence. But in the movie, what can we use as a control? When we hear a curse word, what can we use as a control condition? We want to compare curse words, brain activity for curse words, with something else. All of functional brain imaging is comparison. What's the brain doing now compared to what it's doing at a different time, whether that's a restful period or whether it's some other period. So all brain imaging is comparison. So when you hear a curse word, we want to compare it to when you don't hear a curse word, but is that good enough for us to understand what's going on with curse words? Or should we compare it to words that are matched in arousal, in valence, in frequency? So these are the questions we're facing now. This is not published except at the Neurobiology of Language meeting as abstracts. So we're still working on this. And here's some preliminary data. It took high-performance computing a long time to do this. But we have preliminary data. When people have studied valence all by itself, we get some activity that looks something like what we're getting for curse words compared to, I even forget what control we use. We've tried a number of controls. One control that doesn't work, actually, is when you compare a curse word to periods in the movie when there's no words. What do you expect is going on when there's no words in a movie? In this movie, there's gunshots, there's violence, there's music, arousing music. So it turns out comparing curse words to that doesn't control for valence and arousal. So we're having a little bit of trouble with that. But you can see that there's some good preliminary data here. And here's this is the moderated by arousal. The other was moderated by valence. We did a linear equation. So this is sort of where I've been going and where I've come from, and task-dependent and metalinguistic work and static work. I'm starting in early work on focal regions, and now coming up to today where we're doing movies and ecological kinds of stimuli, real world kinds of things. The ideal thing, if any of you want to do this, the ideal natural experiment is have someone in an MRI scanner in Santiago and another one an MRI scanner in Viña del Mar or wherever, and have them talk to each other while the scanner's going, and record what they're saying, and then code that. That's real natural speech, although the scanners makes a lot of noise. Have anyone been in a scanner? Yeah, it's bad. Okay, so neurobiology of language is possible and depends on understanding language use in the real world. Grasping phenomena such as sentence comprehension and other large-scale language structures will require integration of space and time. Language has been built evolutionarily on sensory, motor, social, and emotional systems of non-human primates. Brain regions perform multiple functions. No function occurs in one location. And language functions appear to organize in core and peripheral regions of the brain with dynamic organization in the face of task demands. And these are the folks who did it all. And they're from all over the world. Patrick is now the director of a neuroscience institute in Hong Kong. Anjali's in Zurich. She's from Turkey, but she's working in the United States. She's in Germany, in Lübeck. She's in the UK. He's in California. He's in London, et cetera, et cetera. And they're really great. Pascal is in Quebec City and is really great. So they did the work, and I thank them. And thank you for your attention. Before we go, I am a gringo, and I do marketing. Here's a good book. It's a nice book. And I don't want any of you publishing in Frontiers. Frontiers is for-profit, charge you thousands of dollars. They take all that money, put it in their pockets. This is $850 for members of the society, $1,100 for people who are not in the society. Costs $100 to join the society. You guys are computer people. You got that math, right? No special formatting, double anonymized reviewing, gender balanced editorial board. My co-editor is Kate Watkins from Oxford. Gold open access, and I really hope people will publish in our journal. And that's it. Thanks. And I'm happy to take questions or comments or critiques. Yes, sir.
Thank you very much for this inspiring talk. So you spoke of localization as simplistic, to which, of course, I agree. Would you comment on that line of work by Jack Gallant at UC Berkeley, where he does this brain dictionary and he has some striking localization results?
Yeah, no, that's interesting. Jack and his student Alex Huth have done a lot of really interesting work on semantic representations. And I don't know what to make of them. They're very inspiring and interesting. He does seem to find some semantic representations, but I don't know to what extent those apply in the same way in a naturalistic kind of environment, whether in a natural environment those grandmother cell kinds of regions actually are larger scale networks when they're actually in use. But it is quite interesting. Yeah, they really are following up on studies that were done by even Wilder Penfield in his neurosurgical work at the Montreal Neurological Institute, and others who have stimulated the inferotemporal cortex in the visual system and evoked images, specific kinds of images. So I'm not sure what to make of it. It certainly is provocative. I like it. I certainly like it. Alex reviews, Jack has reviewed a couple of papers for me for the journals. I like editing, so I do that. And Jack and I know Alex. Alex is in Texas with me, so I know Alex as well. It's pretty good. I don't know. What do you think of it?
Well, you're the expert.
Yeah, I'm not so much the expert at what Jack does. It's it's very much non-ecological. It's very computational, very, very good, really interesting. But I don't know what to make of it in the context of language. He's not studying language. I mean, he's studying, semantic representations of words and items and stuff. But it's cool. It's really cool. Yeah.
Thank you.
Yeah, so thanks for the presentation. And my question goes to the animal model. I think you defined it. Why is it an undergraduate student or teenager?
The animal model.
Yeah.
That's an artifact of how universities are organized. So in many universities, I'm not sure whether that's true in Chile, but in the United States, an undergraduate psychology student has to participate in experiments in order to pass their psychology class. So the professor views participating in an experiment as a way to learn a little bit about how experimental psychology works. So on a college campus, we recruit a lot of undergraduate students, and they participate. We pay them a little bit, but not much, not much. But that's been the history. It's actually, when you read, if you start getting interested in functional MRI research and data, and you look at the literature, take a look at who the population was, because a lot of the results may be more dependent on the population than the authors understand. Suppose you have more men than women. Suppose you have more people of one race than another race. Suppose you have, people with different genetic makeups or whatever. There could be differences. And certainly, trying to understand humanity by a bunch of 20-year-old people is probably not ideal. And that's why, there have been a lot, since the early days, there's different populations that have been studied. By the way, the other advertising I should do, I wanted to do that. I have some brochures here, which I'm going to give to Marcela and Rodrigo, but we have PhD students. So if you are an undergraduate student or undergraduate students, we have PhD programs at the University of Texas at Dallas in all fields that are completely scholarship. We give international scholarships. So someone who comes and does a PhD at the University of Texas at Dallas does not pay for anything, gets a stipend, a living stipend, and doesn't pay tuition. And so we have a lot of those available. In our school, the School of Behavioral and Brain Sciences, we have a Department of Psychology, which does all the human work, including cognitive neuroscience. We have a Department of Neuroscience, which does animal work and in vitro work and some computational work. And then we have a Department of Speech, Language, and Hearing. So I have some brochures which I'll give to the faculty who are here. But if anybody knows someone who'd be interested in that, we are recruiting graduate students. We're also, if you're finishing a postdoc, we are recruiting faculty as well in those three fields. We're recruiting 9 faculty this year, 3 in each of those fields. So if you have colleagues who are looking for positions and are willing to leave Chile, I must say it's one of the most impressive things about Chile is everybody I've talked to says, I would never leave Chile. And I see why. It's really quite an amazing place. But if you know anyone who would consider that, let them know. And we certainly want PhD students to come to University of Texas, Dallas. And I have some brochures for anybody to take with them. So, other questions for me?
Well, thank you. Thank you very much, Stephen. I think when you have shown that the brain, when it's doing language, is so complex, just describing two types of signal. One is hemodynamic, the amount of blood that arrives, and the other is electrical in some EEG study. But we know that we can measure other things. For instance, the balance of neurotransmitter, other type of, even the histology that you have in the brain. So there are many, many, many signals from different type of nature, I mean. So I think in this context, we have a massive data. And this massive data, in order to model this, I think it's extremely important the collaboration of the engineers and mathematicians, or the people is here in the audience. I mean, they really, this work is so big, you know, it's like exploring the universe. It's the complexity is about the same. So what do you think about this multiple domain?
Marcela, I agree completely. And I don't know who's in computer science here, but if you've used convolutional neural networks or other kinds of deep learning devices, those are incredibly powerful tools for analyzing brain signals and trying to understand, infer how the brain's working. And I know some of you are probably looking at the brain as a computation. How does this computational device work? How can I make my AI units, my AI programs, AI systems, work more human-like? I'm not one who's scared of artificial intelligence devices. I'm happy to have them in my hospital. I'm happy to have them everywhere. But I think the model for me in the future is collaboration between artificial intelligence devices and humans. Because right now, your AI devices make errors that no human would make. The reverse is true as well. The human makes errors that no AI device would ever make. And so we need to have that a little bit more convergent. But your tools that you're using, and we use those. We use support vector machines in one study. I think we're using convolutional neural networks in another study, but using these AI methods to infer from data is critical. I once worked with a statistician at Carnegie Mellon when I was in Pittsburgh, and this guy had worked on physics before. He was doing astronomy. And I said, Chris, what do you want to do brain studies? He said, I only care about big data. And if you're a computer scientist and you want to really try to understand big data, there's nothing better than brain imaging studies. And then you're right, Marcelo, with the multi-scale, trying to relate cellular molecular biology, local field potentials, inhibitory excitatory balance, relate those up. Some of you might want to look at my wife's work. My wife, Anna Saladkin, does work with something called the virtual brain. And I would urge you to look that up. It's a brain simulation platform. And again, it's built by computational folks mostly. And it's a brain simulation platform that tries to relate EEG signals and MRI signals down to local fields and inhibitory excitatory balance. And they're even working now in trying to inject drugs into these systems. And the way you inject drugs into one of these mathematical simulations is you say, okay, I have excitation and inhibition in a region in the brain, and I know this drug will affect inhibition or excitation in certain regions. And then how does that propagate through the brain? And then how does it change the MR and EEG signals? And so, I completely agree. The world is now going to require that. Yeah. I lucked out, I trained in computer science and haven't done it forever. But the fact that I trained in it has helped me a lot in my career. So if any of you want to go from computer science into neuroscience, it's not that complicated. And you can join our PhD program with an engineering background. I know my wife would take you in a heartbeat into her lab, but there's a lot of laboratories that really need technical skill. And it's a great background for anything now. I majored in math in school. It's because I was told with math, I was really very versatile, and I have been. It's been good.
Hi, thank you for your talk. I have two questions. One is related with your observation that the resting state, fMRI, changed in some of your interventions. You showed that
Oh, yeah,
after, yeah.
that's right.
So, but my question is whether the resting state before the intervention could predict or may have helped you to, yeah.
I probably explained that poorly, because what I showed here was that the resting state properties before the intervention predicted who would respond to the intervention. That's what I showed you. But I do have a paper on the other. I also have a paper, and this was written by Uri Hassan, who's now at the University of Trento. He was my postdoc in Chicago. And Uri wrote a paper which is published in PNAS, which is a very important journal, on how the resting state changes based on what you were doing before the rest. And in his case, it was a language task. So people were reading stories. And then, so in most of our studies, we do 5 minutes of rest, then we do our study, and then we do 5 minutes of rest. And it turned out the rest at the beginning and the rest at the end were different. And the rest at the end demonstrated aspects of what they were doing right before the rest, the language stuff. So we've used resting state in both ways. Resting state is very popular now in clinical medicine, neurology, and psychiatry, because the people don't have to collaborate with you. You just have them sit there. They don't have to do a task. You don't have task demands. You don't have to have reliability of them doing it. And it's changed. The resting state in Alzheimer's disease is profoundly different than the resting state in healthy adults of the same age. And there are regions that are just operating differently. And when you look at it in terms of network structure, you can say, if you look at the brain in terms of these graph theoretic descriptions, that you have hubs and you have regions that are core nodes and regions that are peripheral nodes, more and less important. Some of the core nodes are just not operating as they should. Some of the big hubs are just not operating as they should. So there's a whole there's a bunch of new theories of Alzheimer's disease, some of these theories are relating the pathology of Alzheimer's disease, the cellular biology, amyloid plaques, beta amyloid and tau, relating some of that to this neural description, the physiology. So it's pretty cool. Yeah, there's a lot of work in resting state now. It was discovered by, I think the first person who wrote about it is a guy named Marcus Rakel, who's a very famous imaging person from the Washington University in St. Louis. And he was elected to our American National Academy of Sciences. And as part of that, he published. Everyone who's elected to that academy, which is the most prestigious honor in America, is invited to put a couple of articles in their journal. And he wrote about the resting state. It's quite, and that was a long time ago. That was probably the turn of the century. So there's a lot of research out there in resting state. Thank you. Thanks, Rodrigo. Thanks, Marcela. Thanks, Alex.